Thank you, Kurt, for that kind introduction. So the title for my talk today is going to be From Microscopes to Satellites, Interactive Image Analysis at Scale Using Spark. So as a brief outline, can everyone hear me, or is it too loud? It's good. We'll talk a little bit about imaging in 2015. So why is this an important theme at all, particularly in research? A little bit about how computing has changed, and then some small data versus big data, and some of the problems we're working on and the solutions we've come up with for them. So for imaging science in 2015, it's really been more and faster. And so if we look particularly in the research area at the Paul Scherrer Institute, where I'm at, the Swiss light source is now producing 8 gigabytes of image data a second. And when you compare this to other places like Facebook or Instagram worldwide, this is substantially more information than they're collecting everywhere in the world. And this is coming from one microscope. So clearly this is a problem to handle this kind of data and come up with analysis tools that can do something useful with it. Similarly, in the hospital or medical area, in Switzerland alone, there'll be more than a petabyte of image data analyzed, or measured. And this is mostly analyzed by eye. And of course, this amount of data is really an overwhelming mass to deal with. And so if you want to study it and improve healthcare and understand what's going on, you need tools that can handle a petabyte. In optical microscopy, there's similar sort of trends that are going on. In geospatial imaging, you see new satellite projects that will make 10 petabytes of images every year. And there's lots of data stuck in these images that someone needs to extract. And even for personal use cases, a GoPro Black can measure 4K video at 30 frames a second, which is already a huge amount of data to try to process on one machine. So if we look at kind of the trends, and this is just from our experience, but it's probably generalizable to a lot of different groups, that if you look at how much data was measured between 2000 and 2016, that it's really grown exponentially. And if you analyze it at kind of a constant rate by looking at pictures, then of course that grows exponentially as well. And so in order to deal with this, you either need to hire exponentially more people or come up with smarter ways for dealing with your problems. If we look specifically in science, and this can also be generalized beyond this field, how much time you spend in each part of the analysis has also radically changed. So when you measure data much quicker, you spend much less time actually measuring it and much more time with the analysis. And so right now we have many examples of students who measured 3 seconds worth of data that they analyzed for 3 years. And so of course to keep up with that is quite intense. And so if we think quickly about how much a terabyte is, as many of you are data scientists, this probably isn't such a revelation, but for physicians it is. If you looked at 1,000 by 1,000 image every second, it would take you 139 hours to go through a terabyte of data. And so if you look at in Switzerland, we can measure that on our machine now in 2 minutes, and you'd need 4,000 people to keep up with it. And that would cost you, 50,000 francs a month. Wait, yeah, something like that. Anyways, it would be very, very expensive, and so you need tools that can handle that analysis. One of the major changes that's happened with computing is that the number of transistors has grown, like Moore's law predicted, but the speed has sort of saturated. So the earlier solutions to problems that you just buy a bigger and faster computer and hope that it works isn't really a viable solution anymore. You need to come up with new kinds of analyses that allow you to distribute and perform these things with multiple CPUs and multiple machines, and that's much trickier than solving it with one machine on one computer. Additionally, if you look at cloud computing costs, the costs have gone down substantially recently, and the cost of buying cloud-based resources versus using a local workstation have gotten so much cheaper that it almost never makes sense to use local workstations because you can just rent computers or as much capacity as you need in the cloud quite easily. So, now to move on, and I'm sure many of you are familiar with this already, so I'll try to cover it quickly, but big data is a very, very popular term to use, and I would say a majority of people who use it don't actually understand it or use it correctly. And so how I would define it is it's not about how much data you have, it's about how you work with it. And so if you have a small amount of data, but you work with it very smart, you're using kind of big data approaches. And if you have a ton of data and you work with it stupidly, you're not using big data. And so here's an example from imaging of what we would call sort of small data. And this is what most researchers are doing, more or less, with their data. And of course, this works fine for one sample, but if you try to scale this up to terabytes or petabytes of images, this becomes incredibly time-consuming. So if we look at a real project that's kind of an example of how this is useful, this genome scale imaging of bones. And so if you want to understand the link between genetic background and the structure of bones, it's important to measure lots and lots of samples. And so with this, what we see that if you only measure 700, it's not enough to establish any substantial links. You need to measure over 1,200 and ideally over 2,000 and even more. And so it's not feasible with standard techniques to perform studies like this. So if we look at this standard small data approach, for 1,300 samples, it would take you 120 man-years. And while the results are interesting, that's really a lot of time to spend on one project and very, very expensive. Of course, you have hand verification of every sample, visual identification of errors and problems, but it's biased, it's questionable reproducibility, versioning your data and keeping track of all your steps is very tedious. So what most groups do, or at least in the research areas, they kind of have these medium data approaches. And this is where you have maybe a bunch of different tools hacked together with scripts and different connections, and this is much faster. So instead of taking 120, it just takes 1.5 man-years to develop this. And it's less biased, more reproducible, but you still have very compartmentalized analysis, and it's very time-consuming to perform. So you have to kind of start each job on its own, and you have to kind of coordinate. This job depends on this one, so if this one crashes, I need to restart that one, or if I want to run it all again, I have to remember to run each step and coordinate all of these things. And so it's quite fragile, and it can be quite sort of expensive. And so before you run the analysis on all your samples, you want to be exactly sure what you're doing, because to rerun it isn't very easy. So yeah, this is sort of the compartmentalized, where you show kind of a time frame that if you decide to change something here, you have to then rerun all of these different steps and make sure you don't forget anything, otherwise you get the wrong results. So we decided that with such a large project, it made sense to really rethink the kind of things we normally do. And so these are what we decided were kind of the priorities for exploratory image processing or image analysis. So the main one we have is, of course, correctness. Because if you do an incorrect analysis, it's meaningless. You're not gathering any new kind of information, and you're just wasting your time. If it's the fastest wrong analysis in the world, it's still wrong. And for that, what's really important is you need to avoid repeating, you need to have things simple, and you need to have everyone be able to understand more or less what's going on. And when you're writing highly optimized GPU code that's doing memory allocation, no one will understand what's going on, even the person who wrote it. And so that's a real major barrier to scaling up. The second is sort of fitting into that, and that's this easily understood, changed, and used, is that we find out with a lot of the samples we analyze, what we thought we wanted to know is very different at the end than it was at the beginning. And so having something where you can quickly switch out parts and change the analysis makes your life much easier and causes you to play with it much more. The last priority there is really fast. And so if you have to wait for 2 days and run a bunch of different scripts to get your analysis, you're not going to explore very much. You're going to kind of do it and say, this is okay, I'm done. And having something that can give you closer to real-time feedback is preferable. So to kind of get into big data a little bit, this velocity, volume, variety, which fits well to our imaging paradigm. When scaling isn't scary, so you can go 10, 100, 1,000 times as big, and it's still the same amount of effort. When you're starving for enough data, so you can analyze everything so quickly that you don't worry about it, and 0 clicks per sample, of course. And so yeah, this kind of summarizes the problems. There's a flood of data. These optimized tools that everyone's been using for a while don't really fit, because they don't scale well, and you need to distribute. You need to use multiple computers to solve the problems. And so what we decided here, as other presentations have already mentioned, was that it was important to use the framework first. So rather than taking the image processing tools and try to figure out how to distribute them, we found a framework which handles this very well and built our tools on top of it. And so what we chose for this was Apache Spark, because right now it has the world record for sorting a petabyte or a petabyte of data. And it can scale beyond 10,000 computers, and you can use it on all sorts of different platforms, and it's very fault tolerant. So if you have 2/3 of your machines crash or get unplugged, it will still finish the analysis. The problem, of course, you have is that Spark doesn't do anything with images. And so if you look at big data frameworks like Hadoop and Spark, they have a big vocabulary, of flat map, filter, aggregate, join, group by, everything else. But none of that sounds like anything you want to do with an image. And so really this important step is translating what you want to do with an image into something that these other platforms can understand. And so that's kind of what we developed with the Spark imaging layer, where you can use the standard vocabulary of image processing, like filter, threshold, component label, shape analysis, and have it run on top of Spark using all of the tools available. So this is sort of the tools we developed at the beginning. So all the different processing and analysis plugins that then fit into the Spark ecosystem. So the purple nodes kind of show what's part of Spark on its own, and the green nodes are everything we developed around it so that you can use it meaningfully in an image environment. So we now look at this genetic study case and with our tools, and this is a mixture of Scala and SQL. Hopefully it's not too scary. You can do everything we did before with several lines of code. And so this takes a study, and it doesn't matter if you have 100 samples, 1,000 samples, or 1,000,000 samples. You read them all in at the same time. You do a filter on them, you segment it, you perform all of these steps as if it were one. So it doesn't become any more complicated, and it's actually easier than most of the analysis are with one sample. Furthermore, you can kind of perform any standard SQL operations on all of these data sets, even though you never actually put them in a database. And so that's one of the advantages of these kind of tools, that you can make very complicated analysis on thousands of data sets that are distributed everywhere, as if it were one simple database, and it's computed at the speed of whatever computers you have available. So, one of the main questions is kind of why hasn't this been done before, particularly in imaging or science, where you see people who are spending lots of time developing new projects. And kind of the main reason is cost. Because if you have 10 samples, spending a lot of time developing big data infrastructure doesn't make sense. It's way more expensive. But as that number of samples starts to go up beyond 10 and 100, it starts to make more and more sense. And so what we see here is that, at 100, it's kind of break even, so medium data is still maybe a little bit cheaper. But once you get to 1,000 or 10,000, then it's a clear advantage. You need to switch over to these platforms that can support this level of scalability, because otherwise you lose too much time managing data and restarting crashed analyses. So, what kind of final tool we've developed out of this is really a quantitative search machine for images. And so I'm sure many of you have used Google image search before. And what we've made is kind of a Google image search where you can actually search for quantitative parameters that scientists are actually interested in. And so you can take something like this map, which is from ESRI's tile data source, and you can say, find all strongly reflective objects, large objects, within 1 kilometer of Paul Scherrer Institute. This can then be translated into a fairly straightforward SQL command that then gets executed on all the different machines, and then you get the results out where you see the structures labeled and the information you wanted to extract from the images. Similarly, in sort of a pathological example, where you're looking at pathology slices from hospitals, you could say, count the number of highly anisotropic nuclei in myeloma patients. This will then be translated into a SQL statement where you run the shape analysis, label the nuclei, do everything else, and export the results. And so what happens there is, of course, it loads in the background all the myeloma data from patients, performs the analysis on every image, and then filters out the anisotropic cells that you're interested in, so you have your final result. And it doesn't matter if there's a terabyte of data or 100 images, it scales and distributes it using whatever resources are available. So here you kind of see how the workflow works, and these slides will be online later if you're interested in studying them in more detail. But basically, you give it this query and it gets handed off to these different components, which then distribute it to, in this case, 4 different machines, and in this case, 100 different machines. And of course, you don't have to worry about how many machines are in the background. It's simply, if you have more, it runs faster. So here's an overview of kind of some of the projects we're looking into and applying these tools for. So for adult zebrafish imaging, to try to study the different cell types, looking at viral infection inside cells, so to see how a time series of images evolves. Looking into sort of personalized medicine and how you can study the connection background and different phenotypes that present themselves. And then the Human Brain Project, which has a small sub-project about mouse brains. For radiology, there's similar sort of applications, with these petabyte of images, trying to test new algorithms and study new sorts of problems. And then here, if you're interested, I can show you a demo where we apply our tools to streaming data so that you can see how you can process images in real time. But I can show that afterwards. So we have a cool tool, and what does this mean for you or other people who are potentially interested? So we've got this quantitative search machine for images that we can apply to your sort of problem. So find all patients with livers larger than 10 centimeters in diameter, or count the number of nuclei, or whatever other task it might be. And we can develop custom analysis solutions based on this framework we've developed. Or as kind of a one-stop shop for doing these kind of analyses. We also provide consulting and education on how these tools work and how you can apply them to your data sets. So I'd like to acknowledge first the people who are part of the spin-off at 4Quant, and then the people from ETH, and thank you for your attention.
Thank you for a very interesting talk. Another application. Are there any questions?
Is this something I can use in open source, or is it something that you aren't releasing? The imaging layer.
So parts of it have been released. The whole imaging layer won't be available as an open source project, but certain components of it will be part of the probably Spark packages. So like the binary support in Spark, we implemented, and we'll probably add some more things to the core that we use, but for like the quantitative search engine, that won't become available.
So I'm not an expert, but I've been just thinking last year at the conference, somebody showed image processing for I think all the satellite images have been released, right, from NASA.
Yeah.
And so they looked at this to study water reservoirs, right, and how like water management, water drainage, et cetera, et cetera. And it seemed that like finding contours, for instance, for water is not actually that easy. So are these image processing filters so standard? Because you're sort of saying, okay, is it so standard that you can have a library of what you do with your image that maps to the standard Spark commands?
Well, the library that we have serves more as giving you the components that you have in any environment to do the image analysis. Of course, if no one has figured out how to identify contours, then our tools probably can't identify contours either. But if you're doing a test for one of these algorithms on 10 images, then you'll probably never find it either. So if you want to learn how to really identify water reservoirs, you need to take, ideally, a data set with the whole world in it and test a bunch of different image algorithms on it to figure out what works everywhere. Because if you just...
You would have the translation of that algorithm into Spark.
Yeah.
Because it's all built out of standard components that you map.
Yeah, so we have standard components, but how you put them together and what exact analysis you build is very flexible. So you can incorporate whatever sorts of analyses you want. We just help you deal with scaling this on 10,000 machines or whatever you need to so you can test it quickly. Because if it also takes an algorithm 2 days to find a water reservoir, you're not going to adjust the parameters because it just takes too long. And if you can easily distribute it on 10,000 machines and each one tries a different parameter, then you're going to try as many parameters as you want because it's doesn't cost anything, or it's very cheap compared to your time.
Okay, maybe one last quick question.
How long is the demo?
I can show the demo now, yeah.
Because in principle, you still have 2 minutes.
Okay, so we can start the demo there.
See, you have extra call already?
So it's starting now in the background, but basically we have this fairly simple outlier detection scheme, and so it uses a webcam image that you see here that's actually coming from my computer. And then we have this median filter being performed to kind of figure out what the background image is. And then if we perform kind of an event that changes the image substantially, it registers as an outlier. And so you can see here that that's an outlier event. And it's processing all of these in real time. And so making it dark was an outlier, and then making it bright again was another outlier. And so if you wanted to detect scene changes or an intruder coming into your house or something like that, you could do it with this system. And really the advantage of this is that you're running a streaming sort of analysis, and right now it's just running on my laptop, but you could run it on 50 machines. And the code for it is simple. I mean, if you wanted to run a different filter or average over 10 seconds instead of 3 seconds, you just change it and click go. And it's, 30 lines of code that covers everything you want to do with these images. And so it really makes it much simpler to deal with these complicated tasks. And if you have 5 cameras instead of just 1, you just have 5 inputs instead of 1. It's very easy to work with. So I think it's still...
Okay, I would say let's thank our speaker again.